You are audible. Okay.
Okay. Good morning, everyone, and welcome to the fifth and final day of the international webinar series, BioSpectrum 2021, on the recent trends in biological sciences, organized by the Postgraduate and Research Department of Microbiology, Sri Shankara College, Kaladi. Today, we are having the technical session 5, led by Dr. S. Unnikrishnan, on mice models in cancer research. Dr. Unnikrishnan has completed his master's degree in microbiology from Sri Shankara College, Kaladi, and his PhD from Sri Chitra Tirunal Institute for Medical Sciences and Technology, Thiruvananthapuram. He worked as a postdoctoral fellow in the Karolinska Institute, Sweden, and worked as a postdoctoral researcher in the University of Oxford, UK. He is currently working as a DHR re-entry fellow in Cochin University, Kerala. Before we begin, I request all the participants to mute your microphones and turn off your video for the smooth conduct of the session. And I would like to welcome Dr. R. Manjula, Associate Professor, PG Department of Microbiology, to welcome the resource person. Madam, please.
Good morning, ladies and gentlemen. Welcome back. I've been assigned a very pleasant duty of welcoming all of you to this platform. I'm proud to introduce today's speaker, Dr. Unnikrishnan, who is a former student of the Postgraduate Department of Microbiology, Sri Shankara College. Started his career as a guest lecturer in Sri Shankara College, Kaladi. He secured his PhD from Sri Chitra Tirunal Institute of Medical Science and Technology, Trivandrum. He worked as an RA at Indian Institute of Science, Bangalore, following which he worked as a postdoctoral fellow at Karolinska Institute, Sweden. He also worked as a postdoctoral researcher at the University of Oxford, UK. He has 13 years of research experience in the field of stem cells, tissue engineering, and cancer biology. At present, he is the DHR Re-Entry Fellow at Cochin University of Science and Technology. He has several awards, and to his credit, he has qualified the National Eligibility Test, and also he was awarded the Junior Research Fellowship instituted by the Indian Council of Medical Research. The Karolinska Institute, Sweden, awarded postdoctoral scholarship as well as research grant to Dr. Unnikrishnan. He was awarded the DHRI HRD grant for rupees 76.5 lakhs in 2021. He has several publications in reputed international journals with high impact factors. Dr. Unnikrishnan, on behalf of the Department of Microbiology and on behalf of Sri Shankara College and on my own behalf, I extend a very warm welcome to you. I welcome my colleagues, students, and also delegates from other colleges who have come to attend this webinar. Once again, a warm welcome to all of you. Have a nice day. Thank you.
Thank you, Madam. Dr. Unnikrishnan, the dais is all yours.
Thank you, Krishna, and thank you, Manjula Miss, for your kind words. I'll start my presentation. Is it visible?
Yes, sir.
And in full screen?
Yes, sir, it's in full screen.
Thank you. So, this is a wonderful experience, presenting in front of my teachers and other colleagues. I did my PhD in Sri Chitra Tirunal Institute, where I was working on in vitro culture models. So always you do cell cultures in tissue culture plates or some matrix. You never get a chance to do something in the animal. And when I did all this stem cell research, I thought to study it completely, you need to have an in vivo system, an animal model. That's how I reached Karolinska, and I was fortunate to get some experience in cancer biology, and my research career shifted to cancer research. So I'll be presenting some of the basic concepts in mouse cancer models, and also I'll be sharing some of my results where I did the research in Karolinska and in Oxford. And my main aim is to introduce these kind of mouse models into the new coming research generation. So I would like to have an interactive session. If something is not clear to you, or if you would like to know more about that model, please raise your hand, stop me, and ask the question. I will try to answer it at my best level. If not, I will get back to you in the future. So please ask questions. Sometimes I will be going into the details without telling the basic mechanism. So you need to stop me to ask the detail. So I wanted to introduce some models, including chemical and radiation-induced cancer mouse models, transgenic mouse models, and under transgenic mouse models, different models like constitutive expression models, conditional transgenic animals, animal models created with RNA interference technology, also with engineered nucleases like CRISPR, and the upcoming models like patient-derived xenograft models and human mouse models. So when I say mouse cancer model, some of you may not even see how it looks like. So I just put some photos. This is what you should expect when you start doing some mouse research. On the left, if I use my laser pointer, on your left, what you see is a neoplasia model. Like it's not a cancer model, it's just a tumor model with papillomas or neoplasm. And if you look at the middle one, what you see is a ulcerative squamous cell carcinoma created with chemical carcinoma. Chemical cancer. It's like, what do you call, the chemicals which can induce cancers. Then far to the left, you can see a transgenic mouse model. So here you are not using any chemicals, you are using some molecular biology techniques to create a mice which gives the cancer. So you can see a breast cancer model here. And on the bottom left, what you see is a cancer formed on a nude mouse. And this is a patient-derived xenograft model. So here these squamous cell carcinoma cells are collected from the patient and introduced into a nude mice, and you get the same kind of cancer in the mice. And bottom middle, you see a melanoma model where you have the abnormal growth of melanocytes. So you can see the black color here because of the melanin accumulation. And onto the bottom far right, you see a orthotopic implantation model. I'll explain all these models in details, but I just wanted to introduce these are the different kinds of animal models. I hope you have heard about ongo mouse. If you not, hear it from me. Ongo mice is the first animal to be patented. And this happened somewhere around 1981. And I would like to divide the research, cancer model research, into two, like before ongo mouse and after ongo mouse. So what was happening before ongo mouse? So the cancer studies were hugely dependent on the cell line cultures. So you collect the cancer cells from the patient, you grow them in vitro on tissue culture dishes, and you study the molecular mechanisms, and you screen different molecules as a therapeutic agent. So it's solely dependent on the in vitro culture conditions. It has some limitations because in vitro you cannot mimic the in vivo condition with as you know, the cancer is a heterogeneous population of cells. It's not made up of one single cell. There will be cancer cells, there will be stromal cells, cancer-associated macrophages. It's a huge different population of cells. So you cannot mimic that in the in vitro condition. Then there was another model where you get these cells from the patient, you grow them in vitro, and you implant into a mice. So this is called the xenograft model. And you can also use some chemical agents or ultraviolet light or radiation to get some DNA damage, which might leads to a cancer. So these were the different techniques researchers were employing before the onco mice. But the introduction of onco mice totally changed the field, and now you have mouse models for each type of cancer. You will see the details in the coming slides. So here I just wanted to give some examples for the chemical carcinogenesis. If you look on the extreme left, you can see different groups of chemicals which can give rise carcinoma. And the mechanism of carcinogenesis hugely depend on the DNA modifications. So if you look at the mechanism of action, either it's a formation of adductive DNA or some DNA breaks or some dimer formation. So everything is dependent upon the DNA damage, and you cannot replicate the exact mechanism for like you cannot reproduce the results if you use chemicals. Sometimes it affects the DNA in some cells. The next time it will be affecting some different cells. So it's a crude methodology. And the other disadvantage is that the chemical carcinogens are carcinogenic to the humans also. So the researcher is under risk. If you are using a chemical carcinogen, you have to take extreme precautions. Otherwise, you will also get cancer. So all these disadvantages was like the researchers was doing some kind of DNA modifications in animal models to get rid of all these limitations. That's how we came to the onco mice. So here I put a few pictures of researchers from 1980s to 1983 to 84, and these were the people that got this idea of creating transgenic models. On the extreme left, Richard Palmer and Ralph Brister, they created an animal model, and they succeeded in getting a brain tumor. And what you see in the right, this white mice is actually the onco mice, what we saw in the previous slide. And this is the first mice to get patented, or first animal to get patented. And on the right, you see a bone cancer model developed by Erwin Wagner. So this was like a huge burst in research. After that, the cancer research is solely dependent on the transgenic. So before going into the details, I would like to explain few terms, like technical terms, which will help you to understand the models in detail. Maybe you already knew all these from your genetic class. So what I wanted to introduce is two kinds of mutations: loss of function mutation and gain of function mutation. For example, if you create an animal and you do some manipulations to get a mutation, and if that mutation creates a loss of gene, or it inhibit the function of gene, then it's called a loss of function mutation, and that kind of mice is called knockout mice. And this loss of function mutation is mainly dependent on the tumor suppressor. Like if you have P53, which is a tumor suppressor, and you create a mutation in P53, and the function of P53 is lost, then you will get a carcinoma. Otherwise, it can be a repressor of a developmental pathway. For example, hedgehog signaling. You maybe already knew about hedgehog signaling. It's an important signaling for development and stem cell differentiation. For example, if you delete the patch 1, which is a repressor of the pathway, if you delete that patch 1, the pathway is constitutively expressed, which also leads to cancer. So these kind of mutation is called loss of function mutation, and those kinds of animals are called knockout mice. And the other type is gain of function. For example, you create a mutation, then that gene is activated. It gives a new function. In my research, I was using a lung cancer model, KRAS G12D. So KRAS is a tumor. It's an pro-oncogene, and the mutation from G to D creates a new function, which gives rise to tumor formation. So those kind of mutations are called gain of function mutation. Then, according to the Mendelian genetics, you know there are homozygous and heterozygous mice, because all the eukaryotic organisms, all the animals are diploid. So we have two alleles, and there will be homozygous and heterozygous mutations. What is heterozygous? You know, one of the allele is manipulated. So if you want to have a cancer model with a heterozygous mutation, then that mutation should be dominant. So, and it works as a gain of function. Then if you have a gene which is homozygous, then it should be it will be a recessive, and usually work it as a loss of function. That's why I explained loss of function and gain of function in the previous slide. And there are some other models where as a mutation in one allele, which is a heterozygous, which usually works as a gain of function, it works as a dominant negative in this function. Like the product from that allele is manipulating the product of the other allele and give rise to a cancer model. So those kinds of animal models are called dominant negative transgenic animals. Then if you look at the expression pattern in the animals, you can divide the mouse models into two types of mouse models: constitutively expressing mouse models and conditionally expressing mouse models. So I was working with conditional mouse models, so I'll be talking more about the conditional mouse models, but I'll briefly explain what is constitutive mouse models. So constitutive or permanent, it's like you are altering the gene permanently, starting from the embryonic days. So when you have a zygote, from the zygote till the end of the animal's life, that gene is mutated or deleted. So those kind of animal models are not very much useful, because sometimes it can be lethal. Like for example, I'll tell SOX2. SOX2 is an important transcription factor. So if you are mutating the SOX2 from the embryonic days, the animal will not survive, because this transcription factor is needed for all the development. So those kind of animal models are not useful when you wanted to study the cancer in an adult life. So basically you wanted a conditional model. So what is a conditional model? Conditional model, in conditional model, you can temporarily, like if you want to get a cancer from 8 weeks after birth, you can activate at 8 weeks. So if you are not interested during the adulthood, you are only interested in the aged mice, you can wait till the animal gets 1 year old, then you can start the manipulation. So these kind of animal models are called temporal conditional model. There are spatial control also. Yes. For example, you are studying the cancer from pancreas. You are only interested in the pancreatic cancer. Then you can specifically target the cells in the pancreas. So some researchers may be interested in cancer of liver, so they can specifically target hepatocytes. So these kind of models are called spatial conditional models. So I already explained this. You can spatially and temporally control the model. And these kind of models are useful in carcinogenesis because, as you know, cancer is a progressive disease. You usually get it during your adulthood or old age because of the mutations accumulating in your DNA. You usually get it after 40 or 50 years. That's the trend you see with cancer statistics because of the accumulation of mutation. So these models can mimic that. If you wanted to get cancer in the adulthood or old age, you can mimic exactly that. So how you do a spatial control of expression of an oncogene? So I'll give you an example. Here, what you do is, I just draw a RNA here. You can see there is a promoter. That's what I gave in yellow color. And this is a protein of interest, or the product, what you expect after the expression. So if you give GLI1 promoter into this construct, whenever the GLI1 promoter is active in the cell, you will get this product. So this product can be a fluorescent molecule, it can be an oncogene, it can be a tumor suppressor, it can be anything. So you can give this transgene to a promoter so that when the promoter is active, you get that specific product. If you are interested to study the function of SOX2, you can put the promoter of SOX2. So whenever SOX2 is active, you will get the product out. So this is how you control the spatial expression. So if I wanted to have the mutation in epithelial cells, the skin epithelial cells, I'll put cytokeratin 14. So cytokeratin 14 is only expressed in the skin epithelial cells. So you will get the oncogene expression only in skin epithelial cells. If you wanted to get pancreatic cancer, you put a PDX1 promoter there. So the PDX1 is only expressed in pancreatic cells. You will get the carcinoma from pancreatic cells. So you can target the tissue of interest and you can get the carcinoma out of that cells. So you don't have to create the mutation in all the cells of your body. You can specifically target a tissue. So before going into the carcinogenesis, I will explain some of the applications of these conditional models. So here I would like to show you some examples of the fluorescent tagging. Instead of the oncogene, you are giving the transgene as a fluorescent protein. So if you wanted to study an event associated with proteomic, metabolic, or cellular genetic events, you can use this model. If you wanted to study the migration of cells, you just give a dye to that cell. So whenever the cell moves from one location to the other location, you can visually see using a microscope. So there are animal models, transgenic animal models, where you knock in a reporter. So a reporter can be a fluorescent molecule. It can be anything which you can track. So the most commonly used reporters are fluorescent molecules or bioluminescent molecules. And the most common fluorescent molecules, you know, tomato, YFP, CFP. So there is a huge array of fluorescent proteins. You can use any of them. So when you combine this reporter animal model with a cancer model, you can see how a cancer originates, how it progresses, and how it spreads, because you already gave a reporter to the mutated cells. Where whenever there is a migration, you can see them. Whenever there is a division, you can see them. Whenever there is phagocytosis, you can see them. And you can also lineage trace. So the term lineage trace explains when it divides, it gives two daughter cells. Both daughter cells will be tagged. So even though this daughter cell goes to the brain and this the other daughter cell goes to the bone, you can see both of them because they are tagged. I'll show you an example from my research where I was studying the papilloma formation from the tumor formation from skin. So my aim was, when you have an abnormal proliferation in the skin, uh, if you look at the skin cells are segregated according to their differentiation pathway. So you can see there are some basal cells. Basal cells divides and give some spinous cells, and the spinous cells divide and give granular cells. So there is a hierarchical pattern, and then never mix together. But as you know, cancer is an abnormal growth. So all the control over the cell growth is gone. So my question was, even if you have this abnormal growth, whether these cells can maintain their anatomical location. So whether they are segregated or they are mixed during the abnormal growth or the tumor formation. So I used a mouse model with 4 reported dyes. GFP, here you can see green in color, YFP, yellow, CFP, cyan fluorescent protein in blue, and RFP in red. So when I did this study, what I observed is, even when you have an abnormal growth, you still maintain that clonal origin. So one clone never mixes with the another clone. So if you are interested in this kind of research, you can use this reporter model. And this is a combined model where you use the reporter mice with a cancer mice. So what I have here is a KRAS G12D mutation. So when the KRAS is mutated, you will get a tumor formation. And whenever this mutated cells divide, they will get one of these colors. And one of, as I mentioned, you can study phagocytosis. So phagocytosis, you can study migration, you can study different developmental pathways. I'll show you 3 examples so that you will understand what is the actual potential of these animals. So I hope the video played. Can somebody answer me? Was it playing, the video?
Yes, but repeat it one more time, please.
Yeah, sure. So so now you look at the left image where I wanted to show you how you can study the phagocytosis. So I showed here a hair follicle. I was mostly studying the skin biology. So I was interested to see the phagocytosis. This is a confocal image, live imaging from a different lab where they studied phagocytosis. If you look at the center area, you can see, you can see the apoptosis, and the apoptotic bodies are formed, and it was phagocytes by the neighboring cell. So I'll play one more. Yeah. So there is apoptosis. You can see the blubbing, like the fragmentation of DNA, and these fragments are phagocytes by the neighboring epithelial cells. So this was the first time when somebody showed the keratinocytes can be a phagocytic cell. This was the first report which was published in Nature. So these kind of experiments you can only do with transgenic models. So the second video explains the same thing. Here it's not getting phagocytes, it's dispersed into the neighboring cells. And here it's a developmental procedure. If you read more about hair follicles, you can learned that the hair follicles goes to 3 consecutive stages like telogen, anagen, and catagen. They grow, they rest for some time, then they start regressing. So it's a continuous process during your life. So here they show the stage of regression. If you look at the hair follicle, you can see it's going upward, which means the length of the hair follicle is getting reduced. So you can study different developmental programs with this reporter mice. And I mentioned about lineage tracing. If you wanted to study a cell's future, like what is the function of this cell? How many more daughter cells will be formed from this single cell? And where are they going? What kind of organs they give rise to? So if you have these kind of research questions, you can use lineage tracing. So it starts from a single cell, it divides, and you can track. If you look the right side, you can track the migration of different progenitor cells. So different color means different level of division. The first division will be red in color. The second division where you get 4 cells will be yellow. Then the third division where you get 8 cells will be blue, something like that. So there are computer algorithms. So you just put these cells under the microscope. You start the microscope for live imaging. You can get this kind of information. Okay, so what we already explained is spatial control. So how can you spatially control the expression? If you only interested in hair follicles, you can go there. If you are interested in the blastocyte formation, you can target this. So that's spatial control. So I'm going to explain something about temporal control. So how can you control the mutation in different life stages? For that, you use maybe you already have it in your curriculum, like Cree log system, FLP log system. Still, I will explain the basics of this system. So temporal control is mainly achieved with the help of Cree recombinase or flipase recombinase or tetracycline operons. So these are the main 3 players in the transgenic models. And you can control the expression of these recombinase enzyme with agents like doxycycline, tetracycline, or RU486. So tetracycline is for tetoperon. Cree recombinase is controlled by tamoxifen, etc. So I'll be explaining examples from tetracycline and tamoxifen, where tamoxifen-based systems were my area of interest, and I was using all the animal models with the tamoxifen-based system. So I'll be giving examples from tamoxifen-based systems so that you will get a better understanding of the system. So here I showed the basic mechanism of tamoxifen. For a tamoxifen activation, you need a promoter so that you can target the tissue of interest. Then you have internal ribosome entry point. If you go back to your molecular biology textbook, you will know more about internal ribosome entry site. And this is the mechanism most of the viruses use. So without the help of other mechanism, they can directly shuttle for protein formation. And you have a reporter protein. So a reporter protein can be a fluorescent molecule. It can be any molecule which can be observed. And you have a recombinase enzyme. It can be a flipase enzyme. It can be a Cree recombinase. And you have an agent which can activate the system. So, for example, it can be tamoxifen or hydroxytamoxifen, tetracycline, anything like that. And you have a log site. So what is happening if you don't have any inducer? Like if you don't have tamoxifen or tetracycline, the system looks like that. Looks like this. Like you have a promoter. For example, I have given GLI1. So this is the promoter. So only the cells which expresses GLI1 will get the mutation. And you have a pre-recombinase, and on the other side, on the DNA, you have an oncogene, and it's flagged by a stop codon. So there is no expression of oncogene in normal case, and you have the reporter before the iris in the internal ribosome entry site. So whenever the oncogene express, you will get a reporter protein or something expressed. So you can see which are the cells which already have this mutation. So in normal conditions, you get a after transcription, you get an mRNA where you have GLI1 promoter and the pre-recombinase, and the pre-recombinase will be expressed. You get a protein of pre-recombinase, but without the inducer for hydroxytamoxifen, nothing is happening. So when you have tamoxifen, for example, I have given an injection of tamoxifen, then what will happen? The RNA will be produced, and this tamoxifen will go and bind to the pre-recombinase, and the pre-recombinase is having an estrogen receptor. If you remember the hormone mechanism, estrogen receptors can move into the nucleus because they are based on cholesterol system. They can move through cell membranes. So when you have tamoxifen, it can move into the nucleus. So when they reach the nucleus, this pre-recombinase will remove the stop, and the oncogene and reporter will be expressed. So that's the basic mechanism of this kind of construct. So if you don't have this tamoxifen, you will not get any expression of the oncogene. So it's basically depend upon the tamoxifen injection. So if you are interested at 8 weeks old mice, you give the tamoxifen injection at 8 weeks, then you have the oncogene expression. If you wanted to wait longer, you wait till 10 weeks or 12 weeks, then you give the injection. So it's temporally controlled. So you can spatially and temporally control the oncogene expression. So this can be done as an excision. If you wanted to remove a complete gene, you can do that. It's basically depends upon the log-P orientation. If you wanted an inversion, you can do that. If you wanted a translocation, you can even do translocation. So you just need to make a transgene with a different orientation of these log-P sites. So coming to some research data, this is my research where I was studying the tumorigenesis from different cell stem cells. Give me a second. So my hypothesis was that when you have the same stem cells in different anatomical locations, we call it as a niche, the immediate microenvironment of a cell. So if you have the same stem cells in different anatomical locations, what will be the influence of this immediate surrounding to give rise to form a cancer? So I used 3 different stem cells. What I showed here is the promoter. So LGR6 is the promoter active in green cells. It's a kind of stem cell. And GLI1 is another kind of promoter which marks a different stem cell. And LGR5 is a marker for a different stem cell. So I was targeting 3 different stem cells, 3 different stem cell populations, and they occupy different anatomical locations in the skin. So I wrote here IFE, that's the area you see from your outside skin. So all these parts is inside the dermal area, that's inside the skin. So you cannot visible with the naked eye, you need to have a microscope to see these stem cells. Then what I did is I created 3 different animal models where when LGR5 is expressed, you will get KRAS mutation and P53 mutation. As I mentioned, KRAS is an oncogene. When the KRAS is mutated, they will act as an oncogene. And P53 is a tumor suppressor in physiological situation. When you have a P53 mutation, their function will be lost. So it's a loss of function mutation, and KRAS is a gain of function mutation, because when you give a mutation, KRAS is giving rise to tumor. So it's a gain of function mutation. So I use these two transgene, and I linked with a protein called tomatoes. So whenever these cells are mutated, the mutated cells will be visible as red. And I created the same with two other stem cells. And these stem cells are occupied in different locations. Then whenever you see a tumor formation with from LGR6, you can study where are they going, what niches are giving rise to tumor. For example, you have LGR6 in this area and this area. If you get tumor from only from this area, you can clearly say that some niche factors from here contributes to the tumorigenesis, or some niche factors from here inhibit the tumorigenesis. So you can easily study these factors with further downstream things like flow cytometry, RNA sequencing, single cell sequencing, or proteomics. So you can study those niche areas, and you can say these are the molecules or signaling mechanism which activates in this niche location, which give rise to cancer. So this is a image I took during my research in Karolinska, where we wanted to create a tumor, and the outcome was different. We got a hair follicle. So it starts from the beginning. It looks like a tumor. You get some abnormal growth, and when it grows, then when it reaches here, it looks something like a bulge in the hair follicle, but we were not sure. But after 12 weeks, we got a full new hair follicle. So if you don't have this red color in that mice, you cannot see this phenotype, because you will think that this new hair follicle is actually a old hair follicle. But here, the only new hair follicles will have this red color. So you can see that, okay, there is something happening. So this is the same animal. So we use the an oncogene, patch 1. I will explain the mechanism here. If you look left side, this is not the same model as I mentioned previously with the KRAS and P53. This is a different model where we use patch 1. So patch 1 is a repressor for Hedgehog signaling. If you remove patch 1, it should be a loss of function, and loss of function should be homozygous. So you should remove patch 1 from both alleles. So you remove patch 1, and you also link the oncogene with a tomato. So whenever you have a mutated cell, you will get tomato. That's what you see in the microscopic picture here. So whenever these cells get multiplied, you will get red color. So you can see where are these cells going, what are the different layers they form, what are the different outcomes from these red cells. Here you can see the new hair follicle is red in color, which means mutated cells are giving rise new hair follicle. And when you have LGR6, they don't give hair follicles, they only give tumor. When you have the Hedgehog signaling manipulated in GLI1, you get hair follicles. So this is how different stem cells behave. So you have the same signaling pathway manipulated in two different stem cells, but when LGR6 is manipulated, you get tumor. In GLI1 stem cells, you get hair follicles. So the organogenesis and tumorigenesis is closely linked. So this paper we already published in eLife, and this is a really interesting results we saw. Like we started with tumor models, and we got hair follicle regeneration. And spatial control, instead of using a specific promoter, you can mix the promoter mechanism with the spatial application. For example, here you have a mice, and the inducer, which is a tamoxifen analog for hydroxytamoxifen, instead of giving the tamoxifen into the systemic circulation, you just paste some of the tamoxifen in the tail. Then only the cells in those area is getting activated, and you get a tumor in the tail. So you can also spatially control with a spatial inducer application. So here you can see the tamoxifen applied areas are getting tumors, and you can link this with a reporter protein with YFP. You can see the mutated cells in yellow color. So here you can see all the abnormal growth is from the yellow mutated cells. So you can spatially control the oncogene expression with the inducer application at specific sites. Okay, this is another combination approach where the researchers use two different mechanisms, like a chemically induced carcinoma with a transgenic animal, a reporter animal. Here what they did is they gave DMBA, which is a chemical carcinogen, with into a mice where they have the confetti. Confetti mice is having 4 different colors. It's a researcher based name, confetti. Like they give the name confetti. It was actually called as rainbow. Then they did some modification. Now it's known as confetti. Confetti is having 4 different colors: GFP, YFP, RFP, and CFP. And when these animals are treated with DMBA, they get carcinoma formation. And all the newly formed cells will have different colors. So what you see on the right side is these are the cells from the tumor, and each of these cells is having a different color. So if you want to study what is coming from the green cells, you can just take different time points, and you can see, okay, green cells are giving rise to the tumor stroma, while red cells are giving rise to endothelial cells, and the yellow cells are actual cancer cells. So you can study the future and fate of different cells. I'll briefly explain tetracycline based system. Even though I haven't used this system in my research, it will be better if you have some overview of the system. So there are 2 kinds of system: tetracycline off and tetracycline on system. Tetracycline off, which is the normal system, widely accepted system. When you have tetracycline, the transcription is switched off. That's why it's tet off. So for this tetracycline system, you have a tetracycline. You should have a tetracycline transactivator, and the inducer here is tetracycline. Instead of tamoxifen in the previous animal model, here you will use tetracycline or a doxycycline. And you have a tet operon, the operator. I'm sure that you already learned the operon systems. Then that's the same system we use here. And when you give doxycycline, the transcription will be stopped. When you remove the doxycycline or tetracycline, you will get the oncogene expression. For example, you have a mice where you have the transgene incorporated, like for example, KRAS. Then you are giving the tetracycline from the embryonic days. You are giving tetracycline in the drinking water or in the food to the mother when she gets pregnant. Then after the pregnancy, the litter will also drinks the same tetracycline or eat the same tetracycline. And after a certain period, you think that this is the time where we should have the oncogene expression. You just remove the tetracycline from the water or the food. You give the normal food, then they start giving rise to cancer. So this is another good mechanism. It's not user friendly, but still you can get the tumor formation with the controlled intake of tetracycline. And this is actually difficult. Like instead of removing tetracycline, you can give tetracycline to induce the expression. That's how the researchers created their own system. So now in normal case, there is no expression of the oncogene. When you give tetracycline, you get the expression. It's just like the tamoxifen system. So you don't have to give the animal tetracycline for a longer time. You can just give the 1 dose or 2 doses of tetracycline, you get the tumor expression, the oncogene expression and tumor formation. And for that, you use a same operon with a reverse transactivator. Okay, coming to the other models, cancer models with RNA interference and other endonucleases. So here, instead of knocking down, if you're only interested in a transient blockage of expression, you can use siRNA. For example, you are studying a metabolic program associated with a cancer formation. You only wanted to have this expression for a short duration, like few minutes to hours. Then you don't have to use the complicated cancer models with transgenes or something. You can just use siRNA. You give the siRNA. During the transcription, siRNA will go and bind to the mRNA, and there will not be any expression of that particular protein. So you can study what is the function of this protein, how this protein play an important role in the cancer formation. So if you are interested in these kind of questions, you can use RNA interference. And RNA interference can be done with siRNA or shRNA. For example, if you wanted to have a longer duration of transient expression, you can use shRNA. If you only wanted few minutes, you can use siRNA. Then comes the engineered nucleases. Nowadays, if you go to any seminar, what you hear will be mostly based on CRISPR associated systems, CRISPR-Cas9, genomic DNA screening with CRISPR. So this is the upcoming animal models with CRISPR systems. So where you give a guide RNA, and the guide RNA will go and bind to the particular gene of interest, the locus. Then the caspase will go there and bind, and you can even modify the genomic sequence with the help of CRISPR. So there are 3 different enzymes available for research, like zinc finger nucleases, transcription activator like effector. It's called a TALEN. Like when you call CRISPR, this one is known as TALEN. But the drawback of these systems are like the efficiency and the cost. You have to create your guide RNA. You have to have this the entire system developed for your studies, and because it's in a new field, most of the research is still based on the transgenic animal models without the use of CRISPR systems. Okay, so coming to the mouse xenograft models, this is another important area in cancer research. For example, when I did my PhD, I was thinking like, okay, to study it in more detail, you need an in vivo system. Then I moved to mouse models. When I did like 4 to 7 years research in mice models, I realized that okay, animals are totally different from humans. So whatever you study in animals cannot replicate exactly into humans. So I started thinking of how can you do some research in humans. So that's not possible. You cannot take a human patient and give some drugs and study what is happening. So the only thing is that you modify the animals to a human system. So here come these models, mouse xenograft models and patient-derived xenograft models. So I will explain these models in detail. So if you are interested to study the exact mechanism or the immunology of cancer formation or the patient-specific drug formulations, then you should have an animal model which is humanized, which more or less mimic the human situation. So in patient-derived xenograft models, when the surgeon removes the cancer during a surgery, you get a small portion of the cancer. You mince them, you make single cell suspensions, and you culture them in vitro, and you inject them into an animal. So this is called patient-derived xenograft model. So actually, the cells are from the patient, the cancer cells are from the patient, and you use mice as an in vivo system to grow them. And there are two types of xenograft models. One is heterotropic implantation model and orthotopic. For example, you take bone cancer cells and you inject them under the skin. So it's not actually mimicking the exact situation. So these kind of models are called heterotropic. For example, you are taking a pancreatic cancer cell and you are implanting directly into the animal's pancreas. Then this will be an orthotopic because both the source of the cell and the donor tissue matches. Then it's called orthotopic implantation. So when you have these kind of research, so when you give the animal human cells, what will happen? There will be some immunological complications. You know, the host versus graft diseases and all these immunological rejection things. So whenever you give an external cell, there will be some immunological problems. To avoid this kind of rejection, you have three different there are different models, but mainly there are three models. I'm sure you have heard about nude mice or skid mice. So the first one is nude mice, where you don't have any functional T cells. Then you have skid mice, where you have no functional T and B cells. And you can have non-obese, that is nude skid mice, where you have no functional T cells and no functional B cells, and without any complications of lymphoma formation. And you have a new variety of mice where you don't have any functional T cells, B cells, and natural killer cells. So you will be removing all the immunological cells from the animal system. So when you give these kind of animals the patient cells, there will not be any rejection. Okay, this is a diagram where you can understand what I was talking about. So you take the cancer and a small piece of that tissue will be implanted into the animal, and the tumor will grow. You take the tumor out. Now it will be 10 times bigger, so that you can give these fragments into 10 animals. Then you actually exponentially grow them in the animals. Now you have a huge number of animals with the same tumor. Now you can screen the drugs. So if you are having 3 candidate drugs, you wanted to see the effect of these drugs against this tumor, you can give these drugs into a group of animals, and you can see some graphs like this. You can see, okay, drug Z is having more effect because the tumor volume is reduced with drug Z. When you have just vehicle like the PBS or some oil, only oil without any drug, you have higher tumor volume. So for these kinds of research, you can have the patient-derived xenograft model. So when I explained the humanized model, I said you will be using nude or nude skid mice, or even the easiest way is actinic mice. You just remove the thymus from the animal, still you will get a mice without any T cells. So any animals where you give the human cells, there will be still some kind of immunological problem, because the circulating blood cells are coming from the animal bone marrow, and the cancer cells are coming from the human patient. So there will be some conflict of signaling. So to avoid that, you can collect the CD34. CD34 is positive cells are the hematopoietic stem cells. So they give rise to most of the blood cells in our system. So you collect the CD34 cells from the patient, you collect the cancer cells from the patient, and you give those to the radiated mice. Where when you radiate the mice, all the bone marrow cells will be dead, and you give CD34. So this CD34 will go and home into the bone marrow, and all the new cells will be human origin. That's why you have to give CD34. So now you have the blood cells from the human patient and also the cancer cells from the human patient. So you can also use these kinds of humanized models. And this is another recent paper where they used a different model. So nowadays, the cancer treatment is moving towards the immunotherapy, where you give the immune cells or cytokines against the tumor, and if you see a reduction, you can use it for further application. So here what they did is they collected the tumor, and there were some tumor infiltrating lymphocytes. So they collected those cells from the tumor, they grow those lymphocytes outside with some media having interleukin-2. Then these cells will expand rapidly in the in vitro system, and you give to the patient or animal again. Then you will see a reduction in the tumor size, because for example, in the tumor there will be only 5 or 10 tumor infiltrating cells, but when you grow them in vitro, they grow exponentially. You get millions of cells. When you give millions of these lymphocytes into the animal or patient, you will get a better outcome. That's what you see in the picture. So even these kind of animal models have some limitations. So I said now we are giving the CD34 cells, hematopoietic stem cells. So the blood cells are coming from the human patient, and also the cancer cells are coming from the human patient. But you know, the cytokines are coming from the animal, and the cytokines are having a huge role on the blood cell differentiation. So for example, if you wanted to have the new natural killer T cell differentiation, you should have a particular array of cytokines, but the cytokines are coming from the animal, so there will be some conflict. So the other way is to engineer some transgenic animals where even the cytokines are of human origin. Then you can avoid this situation. And so when you have all these things, still if you think carefully and logically, you can say that okay, when we give these cells, the T cells will form from the human CD34 cells. Then there will be some complications with the mice, because the T cells are coming from human and it goes into the animal. So there is a rejection from human T cells to the animal cells. But this problem will be solved, because if you give CD34 early progenitor cells, they will be matured in the mice thymus. So you know, mice thymus is the region where you have the double negative selection, where you select the cells which is not acting against your own cells. So this will be solved if you give early progenitor cells. So that the T cells from the bone marrow will go to the thymus, and after the positive and negative selection, all the T cells coming out of the thymus will be acting against human cells, but not mice cells. So I'll conclude my talk. So I hope it was understandable and interesting. So if I conclude, what I was telling, mouse models plays an important role in human cancer research. Most of the questions in cancer research we cannot study in human patients, so we need some animal models to mimic the in vivo conditions. And there are different models available, like chemical models, radiation models, and transgenic models. And the development of transgenic models, the helped in the advancement of research. All the immunological cancer research is now going with the help of transgenic cancer models. And humanized mouse models are the next generation cancer models where we can study immunotherapy and personalized cancer therapeutics. And thank you all for your patient listening. I hope you got something out of my presentation. And if there is something which is not clear, please ask me. And I would like to share this slide. I'm starting a new group. And if you are interested in this kind of research where you study cancer biology in animals and the influence of neurons and blood vessels in the metastasis and cancer formation, you are welcome to join my group. If you are a postdoc, enthusiastic postdoc, or a postgraduate who would like to do a PhD in life science and cancer biology, please send your CV to my email address, unnikrishnan.shivan@yahoo.com. Thank you.
Thank you, sir. And that was an excellent presentation. And now it's time for the query session. And I request all the participants to post their queries in the chat box, or you can directly ask by unmuting your microphones.
Sir, I have one query.
Yeah, could you start with your name?
Pardon?
Could you tell your name, please?
Yeah, I'm Julie Jacob. I work at Martinichus College.
Okay.
I just wanted to know. You told that about the tamoxifen model. We can do temporal expression of tumors. That is, when we inject in the tail of the mice the tamoxifen, all tumor is develops only in the tail.
Yeah.
Yeah, how is it possible? Because in the mouse, in the whole body, it will have this reporter.
Mm-hmm.
Yeah, so when we inject it, it will should go to all parts of the mice, right? So how can we temporarily express that tumor in particular tissue or part of the body?
Julie Jacob, you are now actually talking about the spatial expression. It's not a temporal expression, right?
Yeah, spatial expression.
The spatial expression. So the spatial expression you can either do with a promoter, for example, a particular promoter which is only expressed in the target tissue. As I mentioned, if it's epithelial cells, you use cytokeratins. If it's pancreatic cells, you use PDX1. If it's hepatocytes, you use some liver enzyme promoter. So you can spatially control. Here, what they did is that instead of giving an injection in the systemic circulation, so what you usually do is, at least in my research, what I did is you give tamoxifen to the peritoneum or into the tail vein. So the entire circulation will be filled with tamoxifen, and all the cells which have this promoter will be mutated. But in the system, what they did is, instead of giving into the systemic circulation, they just pasted a small volume in the tail, so that it's just enough to get the cells in that area mutated, but not enough to go into the systemic circulation and mutate other cells in different parts of the body.
Oh, okay, sir. Okay,
So
sir. Thank you.
Yeah, this is mainly controlled with the spatial painting, you only paint a little termostation on your tail so that it's just enough to get the mutation in that area, but not enough to get into the circulation, into the blood, and circulating in the other areas and get the mutation. So that's how they spatially control.
Okay, sir. Thank you. Okay, thank you, sir.
Yeah, thank you.
Hello.
Hello.
Good morning, sir. So my name is Anjali, and I have a question. Like, if a gene it have a normal physiological function in a human body, and if it's mutated, how this technique is we will be able to imply this technique to know whether this gene is mutated or not. A normal functioning gene, if it's mutated, say in case of P53, how will we know whether it have lost its function or not?
Yeah. So, good morning, Anjali. I understood your question. You wanted to see whether these cells are actually mutated. Whether
Yep.
rather than seeing a color in the cell, you actually wanted to see whether they are really mutated. That's your question, right? And also, how to study the function of that mutated gene. Yeah. So, so I'll break these questions into two parts, and I'll answer the first part. How will you find out whether the gene is actually mutated? For that, you have to do a PCR. So it's a kind of genotyping PCR. You make a primer which specifically targets to the mutated area. You do a PCR, and if you see a product, then you can say that there is recombination. So whenever you create a mice, this is the first experiment you should show. You should show a PCR product and show them, okay, this is the mutated area. So you can even take the PCR product, give it for sequencing, and you can show, okay, the sequence shows this mutation. So that's the answer of the first part. The second part, how will you know whether what is the function of the mutated gene? So that's basically on the phenotypic characterization. So you look for some abnormality, you look at the molecular level and also at cellular level, whether something is not right, like whether the cells are not growing, whether it's not dividing, whether this is staying in a particular cell cycle phase, whether it's migrating abnormally, whether it's growing abnormally. So you have to screen the phenotype with some cell molecular biology techniques. So that's how you study the function of the gene. You remove that gene or mutate that gene so that its function will be deleted, and you see what is the effect in the normal physiology. Then you can tell, okay, this gene is having this function. When you delete that gene, this function is not happening.
Okay, thank you, sir.
Correct, Anjali? Now clear?
Yeah. Clear, sir. Thank you.
Okay, it's clear. Okay.
Hello, sir.
Yeah.
So this is Anisa from Shankara.
Hi, Anisa. How are you?
Fine. Actually, sir, I want to know that you were describing about lineage tracing, where you were saying we can use multiple reporter molecules to track the hierarchy where it is actually going and located in future, right? Okay. Actually, all these kind of reporters we are introducing into a single cell, right?
No.
No.
That's the only question. Or you have something.
I just want to know, because we are doing all this modification in a particular cell. That's what I understood. In a particular cell, when it goes division, undergoes division and differentiates into different type of cells. We can trace to which part actually, what kind of differentiation is actually taking place, and where it is exactly going and locating in future. That's what I understood. understood. But my query is how such kind of modification can means these four molecules. You are describing 4 reporter gene, 4 or 5 reporter molecule. How this can be introduced into that particular study means at its initial stage.
Anisa, thank you for the question. That's a wonderful question. Fortunately, I was prepared for this question. I will share my screen again and I will explain this model in detail. So before going into that slide, the first question, whether this is happening in a single cell. So that's actually not correct. This is not a single cell manipulation. When you have a particular promoter, you are targeting a population of cells. A cell population which is having same function and same promoter expression. For example, if you are studying skin and you are targeting with some keratin protein, for example, I'll use CK5, then cytokeratin 5 is only expressed in a population of cells which resides above the basement membrane. So it's not a single cell, it's a population of cells. Whenever you do these kind of manipulation, it's a population of cells. So it's not a single cell. Now I'll present my screen again. Yeah. Can you see my screen?
Yeah, it is.
Anita, so I gave 3 boxes here. The first one is construct, the second one is outcome, and the third one is what you see under a microscope. Look at the construct. If you go back to this slide, where I was little fast, so you can modify the pattern according to the orientation of the log sites. So if you give that same orientation, that protein will be deleted. If you give an inversion, if you give in different orientation, there will be an inversion. If you give a trans orientation, you will get a translocation. So what they did is, again, this is a Nature paper where they created this model, and it was actually called as Brainbow 2. So they incorporated 4 different fluorescent molecules, and instead of giving the same orientation, they gave different orientation. For example, when they you take this one and this one, that's the possibility I, and the possibility I will give GFP, because YFP in the other orientation, so it will not be expressed. So you will get only GFP. Take the second option. You have different orientation of LOXP site here, this one and this one, and that is the second option. In the second option, you will get an, when it's in the different orientation, there will be an inversion. So you will get CFP expression. And so according to these different orientations, you will get different outcomes. Either you get RFP, for example, RFP comes from 3 is like this. You see, if the recombination happens here and here, you get RFP. And this is a probability thing. So when you give tamoxifen, one cell might be recombining in this scenario, one condition, the other cell in the same population will be having the recombination in the 2. So it's a recombination probability. Like when you study the genetics, when you study the crossing over and all with linked genes, you learn about the probability, right? So it's basically depends upon the probability. You will have 4 outcomes at different levels. And when you have a single cell with a red dye and all the cells coming out of this cell will be red in color. So you can track them clonally. So from the starting, you will have 4 different clones with the CFP, RFP, YFP, and GFP. After few generations, there will be a clone, like a clump of cells. That's what I showed in this slide. From the starting, it was a single CFU, like a clone forming unit, a single cell, which was having the recombination with the GFP. And after some time, this clone expanded, which gives rise to a clump of cells. So that will be green in color. If these clones are mixing together, you will get green and yellow cells in the same region. So you are targeting a population of cells, and in that population, when you have 4 different molecules, each cell can get different colors. And when after the treatment, you take a picture and you see, okay, these are the starting recombination findings. Then you grow them for some time and you look again under the microscope and see, okay, now the red clone overgrown and the yellow clone is totally lost, and the blue clone forms the external layer of the cancer, and the other clone forms the blood vessels. So you can actually study their fate. Anisa, is it clear now?
Yeah, of course it is clear now. Thank you.
So if you want to learn more about this paper, just Google Brainbow 2 and you will get this paper, and they explain nicely with all the mathematical calculations for the probability of each color, and you can easily understand it. Is there any other questions?
Hello, sir. I think there are no other questions.
Thank you, Krishna.
So thank you, sir. Thank you, sir, for answering the questions of the participants. And I request all the participants to find the feedback form provided in the chat box and to fill the form as well. And the e-certificates will only be generated after filling all the 5 feedback forms. Now I welcome Mr. Vinayak V, student representative, to deliver the word of thanks. Vinayak, please.
Am I audible?
Yes, you are.
Hello, am I audible? Okay, good morning, everyone. Today, we have came to technical session 5 of the BIOSPECTRUM 2021 International Webinar Series, organized by the Postgraduate and Research Department of Microbiology, Sri Shankara College, Karali. And the duty vested with me is to give the vote of thanks. First of all, I would like to thank today's resource person, Dr. Unnikrishnan, who gave an exemplary presentation on mice models in cancer research. And I believe the session was well informed and effective. Thank you, sir. And I thank all the participants for your kind cooperation. And I thank all the members who have worked behind the coordination of this session. Once again, I thank everyone. Thank you. Stay safe.
Thank